Hi, so today is the ninth problem solving session for the course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goel, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. So today we'll be discussing certain topics and questions related to those topics, and these questions will be similar to the questions given for the assignment of this week. And feel free to ask any questions if you have. Let me know if you have any doubt in any of the questions discussed here or in any of the topics that are there in this week's lecture series. So I'll start with the questions. Just ask your doubts in the chat box, or you can unmute your mic and ask. Okay. I hope everybody can see my screen and I'm clearly audible to everyone. If someone can respond in the chat box. Okay. Sure, then I'll start. I'll switch on the switch off the video to save the bandwidth. Mm. Okay, so starting with the first question. Which of the following terms is used to describe the increase in concentration of a chemical compound in the tissue of a living organism with respect to water? Which of the following terms is used to describe the increase in concentration of a chemical compound in the tissue of a living organism with respect to water? Option A, live accumulation factor. Option B, bioconcentration factor. Option C, chemical accumulation factor, or option D, chemical concentration factor. So, what is the correct answer? If anyone knows the answer, write it in the chat box. I request all of you to try to answer the question. Even if you don't know the answer, try to think logically and try to answer so that we can have a discussion. Anybody? What would be the correct answer to this? Come on, write it in the chat box. It's okay if your answer is wrong here. We can always have a discussion whether it's a right or wrong answer. Just answer. Okay, we have one answer in the chat box, option B. Okay. Anyone else wants to answer? Okay, let's see the answer. Option B is the correct answer, bioconcentration factor. Very good. So, bioconcentration factor, as you can see in the question, it is basically the amount of any chemical compound. So, these chemical compounds are generally synthetic compounds which cannot be degraded and they remain in the environment for a long time. So, they get accumulated in our tissues also, okay, in our or any living organism, okay. So with respect to water, the concentration of particular compound in water and the concentration of the same compound in tissue, that comparison, like that ratio, is basically defined as the bioconcentration factor. So it is the ratio of the concentration of the compound in the tissue of a living organism with respect to the concentration of that same compound in water. Okay, so it is generally written as this. So C biota here denotes concentration of the compound in the living organism. So in any biotic, in any living organism or any biota, the concentration of a particular compound and the concentration of the same compound in water. Okay, so that is how we define bioconcentration factor. Okay, and this is a very important factor, because if uh, any compound has a higher bioconcentration factor, that means it is there, it is accumulated in high amount in the living organism, in the tissue of a specific organism, and that can be a health hazard. Okay, so that's why this factor is very important. We need to determine this factor for every chemical compound, so that we can determine whether that particular chemical compound is safe or not. Okay, that's clear to everyone. If you have any doubt in this, write it in the chat box, or you can unmute and ask. Hope that's clear to everyone. I'll move on to the next question. Question number 2. Which of the following is not a xenobiotic compound? Which of the following is not a xenobiotic compound? Okay. So option A is dichloro-diphenyl trichloroethane. Option B is trinitrotoluene. Option C is glutathione. And option D is fluorine. So we have 4 options. First is dichloro-diphenyl trichloroethane. Second is trinitrotoluene. Third is glutathione. And fourth is fluorine. So you have to tell which of the following is not a xenobiotic compound. Okay. So write your answers in the chat box. Which of the following compounds do you think is not a xenobiotic compound? Anybody knows? Can anybody answer which compound is not a xenobiotic compound? So do you know what a xenobiotic compound is? So basically, xenobiotic compound is a synthetic chemical compound, which is synthesized. It is not present naturally. It is not present in nature. It is not found in any plants or animals or anywhere. It is synthesized chemically by humans. Okay, so that those are xenobiotic compounds. So they are generally, since they are chemically synthesized, they are generally non-biodegradable and all. Now tell me, which of the following do you think is not a xenobiotic compound? So 3 of them are xenobiotic compound, only 1 of them is not. Come on, write your answers. Nobody knows the answer. Nobody wants to even try to answer. Okay, so I'll tell you the answer. The correct answer is option C, glutathione. So, glutathione is naturally available compound. All other compounds are xenobiotics. So, I told you xenobiotic is a synthetic organic compound that are relatively alien or foreign to the environment. So, common examples are herbicides and pesticides. These compounds can be hazardous and toxic, and they are often toxic to higher and lower organisms, source of pollution problems, short-term and long-term health effects, depending on the concentrations and routes of exposure. So you must have heard of certain compounds that are used as pesticides, and they are nowadays causing a lot of health related issues since they are used in the uh, vegetable farms where all these vegetables are grown, and we are consuming those vegetables. So indirectly, we are also consuming those pesticides, and they are getting accumulated in our tissues. And that's how the bioconcentration factor also comes into the picture. Okay. So these xenobiotic compounds in the environment are degraded by physical, chemical, and biological reactions. So there are certain methods to degrade these uh, compounds in the environment, but again, it's not very easy to degrade these compounds completely and reduce their toxicity levels. Okay, so in the options we had dichloro-diphenyl trichloroethane, that is DDT. So it is a very commonly used insecticide which is used in agriculture. So DDT is one of the major insecticide that has been causing health related issues since they are used in the agriculture, and we are consuming those agricultural products, those fruits and vegetables that are grown, and we are indirectly consuming DDT, which is a big health hazard right now. The second option was trinitrotoluene. So trinitrotoluene, or in short TNT, it is chiefly used as an explosive. So it is mainly used in the explosives that are used by military and all. Then another xenobiotic compound that was there was fluorine, which is used to make dyes, plastics, and pesticides. The only compound that was not a xenobiotic was glutathione. So glutathione is a peptide, and it is found in plants and animals. It is powerful antioxidant produced by liver to protect the body against free radicals, peroxides, and heavy metals. So glutathione also eliminates poisons such as drugs and pollutants from our body. So it is a natural compound that is produced by our body. Okay. So it is not a xenobiotic. The rest all the other compounds are xenobiotic. That's clear? Okay, moving on to the next question now. If the octanol by water partition log coefficient value of benzopyrene is 6.06, the compound is practically option A hydrophilic or option B hydrophobic. So if the log coefficient value of benzopyrene is 6.06. This value basically denotes the concentration of a compound in octanol uh, versus water. Okay, concentration of the particular compound in octanol divided by the concentration of that compound in water. So if the logarithmic of that coefficient is 6.06, what do you think the compound is hydrophilic or hydrophobic? Can anyone answer? Write your answers in the chat box. think logically, the concentration of a particular compound in octanol versus the concentration of that same compound in water, if that is given to you, and now I'm asking if the compound is hydrophilic or hydrophobic. Hydrophilic means it is more likely to be soluble in water, and hydrophobic, that means it is not very soluble in water. Maybe it can be soluble in octanol. So this is your hint. Think and answer. This is not a very difficult question. Everybody should be able to answer this question. At least try to understand the question and write your answers. Come on, anyone? Nobody knows the answer. Nobody can think of a correct answer. There are just two options. Okay. Okay, two people are saying it can be hydrophilic. Okay. Anyone else wants to try? Let's see. Actually, the correct answer is hydrophobic. So I told you the value that is given, it is the concentration of a particular compound in octanol versus the concentration of that same compound in water. So now if the value is 6. Log KOW is 6.06. Here I told you log of that coefficient is 6.06. So that means the value of KOW, or the uh, partition coefficient, it is 10 raised to the power of 6.06. That means the concentration of that compound of benzopyrene in octanol is 10 raised to the power of 6.06 times the concentration in water. So that means it is more soluble in octanol than water. Okay, so this value indicates the water solubility of a compound is low as compared to the solubility in octanol. So that means it is hydrophobic. It is less soluble in water, so it is hydrophobic. If the value uh, for this KOW was less than uh, 0, if it was less than 1, then the value would have been, the compound would have been hydrophilic. Okay. So this partition coefficient, so here you can see in this graph, so this is the log of that partition coefficient, that is log KOW, and this shows the bioaccumulation factor. So higher the partition coefficient, that means higher the compound is hydrophobic, chances of it getting accumulated in the environment or accumulated in the living tissue is higher. Why? Because obviously, if a compound is hydrophobic, it is not going to get solubilized in water. It is going to remain in the environment as it is. If it is water soluble, it can get washed off from the surface. But if it is not water soluble, it will get absorbed in the tissues, and it will remain accumulated, because in your body also the environment is hydrophilic, right? So bioaccumulation factor is directly proportional to the log octanol by water partition coefficient. Okay, this is clear to everyone. Do you understand why the compound is hydrophobic and not hydrophilic? Okay. Okay, then I'll move on to the next question. Question number 4. How many intermediates are generated in a glycolysis pathway? How many intermediates are generated in a glycolysis pathway? Option A 5, option B 8, option C 9 and option D 10. How many intermediates are generated in a glycolysis pathway? 5, 8, 9 or 10? Come on everyone, write your answers in the chat box. Okay, I have one answer, option D, that is 10. Okay, someone is saying 5 and then 9. Okay, so I have multiple options in the chat box. Anyone else wants to try? Okay. So I can see people answering 10, 5, and then 2 answers are 9. Okay, let's see the correct answer. The correct answer is actually 10. So there are 10 total intermediates in the glycolysis pathway. So glycolysis pathway starts from glucose and it ends with pyruvate, and in between there are total 10 steps. Okay, so there are 10 different compounds that form before it reaches the final compound, that is pyruvate. So we start with glucose. So the first thing that first compound that is formed is the phosphorylated form of glucose. So the sixth at the sixth position, phosphorylation occurs and we get glucose 6-phosphate, and the reaction is carried out by hexokinase, the first enzyme in the pathway. Then from glucose 6-phosphate, it moves on to fructose 6-phosphate by another enzyme, an isomerase. Then from fructose 6-phosphate, another step of phosphorylation occurs where it becomes fructose 1,6 biphosphate. So two phosphates are attached to the fructose molecule. From this, two intermediates are generated by an enzyme called aldolase. One is glyceraldehyde glyceraldehyde phosphate, and second is dihydroxyacetophenone. Okay, so these two compounds are generated, and then dihydroxyacetophenone. So these two compounds they remain in equilibrium and they can change into one another. So then finally, after this step, after this fifth step, so here we get two intermediates. Then from this step, glyceraldehyde phosphate dehydrogenase converts this glyceraldehyde phosphate into 1,3-phosphoglycerate. So we have two phosphates here in a glycerate molecule. So 1,3-phosphoglycerate. So this dihydroxyacetophenone also gets converted into GADP, and then it converts by GAPDH, it gets converted into 1,3-phosphoglycerate. From this step, we have the next intermediate 3-phosphoglycerate where the dephosphorylation reaction occurs. So this is the first dephosphorylation reaction that is happening. So here you remember we had phosphorylation reactions. So from glucose to glucose 6-phosphate, we had a phosphorylation step. Then fructose 6-phosphate to fructose 1,6 biphosphate, we had another phosphorylation step. Now after this, glyceraldehyde 1,3-phosphoglycerate. From 1,3-phosphoglycerate, we are getting 3-phosphoglycerate. This is a dephosphorylation step where the phosphate is removed from the substrate. Then after 3-phosphoglycerate, the phosphate shifts to the second position by a mutase enzyme, and it becomes 2-phosphoglycerate. And this 2-phosphoglycerate then gets converted into phosphoenol pyruvate by enolase. And finally, phosphoenol pyruvate gets converted into pyruvate. So this is the last step, is the second dephosphorylation step. So we have two major phosphorylation events and two major dephosphorylation events in the glycolysis pathway. Okay, and in total, if you can see, you can count, we have 10 intermediates. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay. So we have in total 10 intermediates in the glycolysis pathway. Is that clear to everybody? This is clear to everyone. We'll discuss more questions related to glycolysis. So if you have any doubt in any of the intermediate steps, we'll have a discussion. If you have any doubt right now, you can write it in the chat box. Moving on to the next question. Which of the following is a crucial cofactor for the reaction in the glycolysis pathway? Which of the following is a crucial cofactor for the reactions in the glycolysis pathway? So you just now saw there are 10 reactions going on in the glycolysis pathway, right? So out of the following four cofactors, option A is Mg2 plus, option B Cu2 plus, option C S2 minus, and option D is N3 minus, that is nitrogen ion. So out of these four cofactors, which of the following is crucial for the reactions in glycolysis pathway? So which cofactor do you think is crucial? You saw the reactions now. What do you think is crucial for the reaction? So I'll give you a hint. The cofactor is actually important part of the enzymes that are functioning in the pathway. Okay, I'm getting option A as the answer in the chat box. Two people are answering option A. What about others? Option A is Mg2 plus. Okay. Okay. I have two more answers. They are also option A. So everybody seems to agree with option A. That is the correct answer, Mg2 plus. So Mg2 plus, that is the magnesium ion. It is an important cofactor in the glycolysis pathway that helps the enzymes hexokinase, phosphofructokinase, glyceraldehyde phosphate dehydrogenase, and phosphoglycerate kinase to function and catalyze these reactions. So total four enzymes: hexokinase, phosphofructokinase, glyceraldehyde phosphate dehydrogenase, that is GAPDH, and phosphoglycerate kinase. Four enzymes are having Mg2 plus as a cofactor. They use Mg2 plus as a cofactor. So uh, if you see in this pathway, so we have here hexokinase, that is the first step of the glycolysis. So from glucose, it goes to glucose 6-phosphate, and hexokinase is the enzyme. So this also requires Mg2 plus. Then phosphofructokinase, which basically converts fructose 6-phosphates into fructose 1,6 biphosphate. So this is the second phosphorylation step. So this also uses Mg2 plus as a cofactor. Then we had GAPDH, glyceraldehyde phosphate dehydrogenase, that was converting glyceraldehyde phosphate into 1,3-phosphoglycerate. So that is also using Mg2 plus as a cofactor. And then phosphoglycerate kinase, that was doing the dephosphorylation reaction, 1,3-phosphoglycerate to 3-phosphoglycerate, that is also using Mg2 plus. So four enzymes are there in the pathway, that out of 10, 4 enzymes require Mg2 plus. So it is an important cofactor in the glycolysis pathway. That's clear to everyone? Everyone's Most of you gave the correct answer. So I hope this is clear to everybody. Anybody has any doubt in this? Please ask. Okay, then moving on to the next question. Which of the following reaction yields ADP from ATP in the glycolysis pathway? Which of the following reaction yields ADP from ATP in the glycolysis pathway? So we had 10 steps in the glycolysis pathway, right? I told you there were phosphorylation and dephosphorylation steps. Okay. So now if a reaction is yielding ADP from ATP, that means ADP is gaining one phosphate to form ATP. What does it mean? That means there's one substrate that is losing a phosphate that is acquired by ADP to form ATP. So we are getting which of the question is which of the following reaction yields ADP from ATP. That means ATP is getting dephosphorylated to form ADP, right? So that means ATP is giving the phosphate to something. So something is getting some substrate is getting phosphorylated. So read the options carefully. First option is 1,3 diphosphoglyceric acid to 3-phosphoglyceric acid. Second option is fructose 6-phosphate to fructose 1,6 diphosphate. Third option is phosphoenol pyruvic acid to pyruvic acid. Fourth option is 3-phosphoglyceric acid to 2-phosphoglyceric acid. What do you think is the correct answer? So which of the following reaction yields ADP from ATP? That means from ATP, ADP is forming. That means ATP is giving the phosphate to some substrate. Which of the substrate here looks like it is getting phosphorylated? Okay, we have 3 answers in the chat box. All of them are option B. Fructose 6-phosphate to fructose 1,6 diphosphate. Yes, that is the correct answer. Option B. So here if you see logical, this is you just have to use logic here. So I told you we are getting ADP from ATP. That means ATP is giving one phosphate. So ATP is adenine triphosphate and this is diphosphate. So from a triphosphate, diphosphate is forming. So that means one phosphate is going somewhere. So where it is going to a substrate. So now look at all these substrates. So the first option, 1,3 diphosphoglycerate to phosphoglyceric acid. So this is dephosphorylation step. So that means it is not acquiring a phosphate, it is losing a phosphate. So this cannot be the correct answer. In the second option, fructose 6-phosphate to fructose 1,6 diphosphate, it is acquiring a phosphate. So this is a correct answer. Third option also, the phosphate is getting lost from phosphoenol pyruvic acid. So phosphoenol pyruvate is losing the phosphate to become pyruvic acid. Okay, so it is donating the phosphate. So in these step, in first step and in the first option and in the third option, in these step basically ADP will convert into ATP. But in the second option, ATP will convert into ADP because ATP is giving the phosphate to the fructose 6-phosphate to become fructose 1,6 diphosphate. And fourth step is just a isomerization step. So the only the phosphate position of the phosphate group is changing. The compound is neither getting phosphorylated or dephosphorylated. Okay. So here you can see this is the step where ATP is ADP is yielded. So there's another step where ADP is we yield ADP from ATP. So here the first step, that was glucose getting converted into glucose 6 phosphate by a hexokinase. So this is also phosphorylation step. ATP is giving the phosphate to glucose to become glucose 6 phosphate, and in turn it is getting converted into ADP. In this step also you see fructose 6 phosphate is taking one phosphate from ATP to become fructose 1,6 biphosphate, and ATP is getting converted into ADP. In these two steps, you can see opposite is happening. So from 1,3 biphosphoglycerate, you are getting 3 phosphoglycerate. So one phosphate is given by 1,3 biphosphoglycerate to ADP to form ATP. Okay. And here also from phosphoenol pyruvate, the phosphoenol pyruvate is giving one phosphate to ADP to form ATP, and in turn it is getting dephosphorylated to form pyruvate. Okay. I hope that's clear to everybody. This is clear to everyone. If you have any doubt, please ask. So this is a very important pathway. So you should all be able to understand the pathway. If you understand the pathway, you don't even have to memorize the pathway. It will automatically be clear to you. Okay. Okay. If this is clear, then I'll move on to the next question. Question number 7.
Ma'am,
Yeah. Yeah, tell me.
clear, ma'am.
Okay. Okay. Question number 7. Glycolysis starts with dash and ends with dash. Now I've shown you the glycolysis pathway in the previous question, so everyone should be able to answer this. Glycolysis starts with dash and ends with dash. Option A, one glucose molecule. So basically glycolysis starts with one glucose molecule and ends with one pyruvate and one ATP. Second option is glycolysis starts with one glucose molecule and ends with one pyruvate and two ADP. Third option is glycolysis starts with one glucose molecule and ends with two pyruvate and two ATP. Fourth option is glycolysis starts with two glucose molecules and ends with two pyruvate and one ATP. Which of the following is the correct answer here? You all have seen the answer already in the previous question. So everybody should be able to answer this. Come on, write your answers in the chat box. Which option is correct? Write your answers in the chat box. Okay, we have one answer, option A, that is glycolysis starts with one glucose molecule and ends with one pyruvate and one ATP. Okay, another answer is option C. Glycolysis starts with one glucose molecule, ends with two pyruvate and two ATP. Okay. Okay, now we have 3 people agreeing to option C. Okay, a lot of people are agreeing to option C, it seems. Yeah, so that is the correct answer. So majority of you are right. So option C, that is glycol, also starts with one glucose molecule and ends with two pyruvate molecules and two ATP molecules. That is the correct answer. So again to uh, as a revision, this is the glycolysis pathway. So it is starting with one glucose molecule. How is it ending up with two pyruvate molecule? This is very simple. So glucose is a 6-carbon compound, right? And pyruvate is a 3-carbon compound. So simple equilibrium will, simple chemical reaction and equilibrium will tell you that this 6-carbon compound will get converted into two molecules of 3-carbon compound. Okay, so that is the simplest explanation. And if you see here, from glucose, we are going to glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-biphosphate. After this fructose 1,6-biphosphate, this is getting converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, so these two compounds, these are getting changed by the enzyme isomerase. Okay, so from this point we are getting technically two molecules of glyceraldehyde 3-phosphate. So fructose 1,6-biphosphate is also 6-carbon molecule. Sorry, it is also 6-carbon molecule. So from this 6-carbon molecule we are getting two molecules of a 3-carbon compound. Okay, so two molecules of 3-carbon compound. So we are getting two molecules of glyceraldehyde 3-phosphate. That two molecules of glyceraldehyde 3-phosphate now will get converted into two molecules of 1,3 biphosphoglycerate, then two molecules of 3-phosphoglycerate, two molecules of phosphoglycerate, then two molecules of phosphoenol pyruvate, and finally we are having two molecules of pyruvate. So this part I hope is clear to everyone. One glucose is getting converted into two pyruvate because glucose is 6-carbon. So if I have to balance the equation, so one glucose will get converted into two molecules of a 3-carbon compound, that is pyruvate. Now let's see what is happening to the ATP. So here in the first step, one ATP is getting consumed. Okay. In the third step also one ATP is getting consumed. Okay. Now, in this step here, you see two molecules of 1,3-biphosphoglycerate is there, and it is getting converted into 3-phosphoglycerate. So, one molecule of bisphosphoglycerate, it will give one phosphate to one ATP, right? So, one ADP. Okay. So, since there are two molecules, there will be two ADPs taking two phosphate groups to form two ATPs. So, in this step, two ATPs are forming. Same thing is happening in the last step. So there are two molecules of phosphoenol pyruvate. They can give two phosphate groups to two molecules of ADP, and two ATPs will be forming here. So now if you see here two ATP is forming, here two ATP is forming. So total how many ATP are forming? 4 ATPs are forming. But here one ATP is getting consumed, and in this step also one ATP is getting consumed. That means 4 ATPs are forming, 2 ATPs are getting consumed. So net number of ATP that we are getting finally are 2. Okay. So 4 ATPs have formed, 2 ATPs are used out of it. So what is remaining is 2 ATP. Okay. So we have finally starting with one glucose molecule, we have finally two pyruvate molecules and two ATP molecules. Okay. That's clear to everybody. People who haven't answered anything or people who have answered other options than option C, is it clear to you now? So glycolysis starts with glucose, ends with two molecules of pyruvate and two molecules of ATP. Okay, I think I can move on to the next question now. Question number 8. Under anaerobic conditions, Saccharomyces species can convert pyruvate into the following. Option A, lactic acid. Option B, ethanol and CO2. Option C, acetic acid and CO2. And option D, butyric acid and isopropyl alcohol. Under anaerobic condition, Saccharomyces species, that is yeast, it can convert pyruvate into the following. So yeast can convert pyruvate into one of the following compounds under anaerobic condition, under anoxic condition. Okay. Option A is lactic acid. Option B is ethanol and CO2. Option C is acetic acid and CO2. And option D is butyric acid and isopropyl alcohol. Okay. I have okay, two people are saying option B. What about others? Okay, four people are agreeing with option B. So that is the correct answer. So you all are correct. Ethanol and CO2. Under anoxic condition, pyruvate can be converted into uh, two different compounds depending on what kind of process is followed. So this is glycolysis. From glucose, we are getting 2 molecules of pyruvate, that is glycolysis, 10 successive reactions. After pyruvate formation, under normal aerobic condition, like in humans also, when we are breathing and we are surviving under aerobic conditions, right? We need oxygen to survive. So in the presence of oxygen will be the terminal electron acceptor. Finally, oxygen is going to accept the electron. So these two pyruvate under aerobic condition, they will be converted into acetyl coenzyme A, and then finally CO2 and H2O. Okay, final oxidation of all the compounds will happen and we'll get carbon dioxide and water, and all the electrons that are released in the reactions, they will be accepted by the oxygen. But in case where the oxygen is not present, so in those cases, there can be two scenarios. So first scenario that occurs in yeast under anaerobic condition, it converts the pyruvate into ethanol and carbon dioxide. Okay, so it uses the enzyme pyruvate decarboxylase, where decarboxylation of this pyruvate occurs. So the CO2 is released and acetaldehyde form. This acetaldehyde then gets converted into ethanol by alcohol dehydrogenase. Okay. So pyruvate decarboxylase, it requires Mg2 plus, and it does the decarboxylation of pyruvate to form acetaldehyde, and then from acetaldehyde, the alcohol dehydrogenase converts it into ethanol. Okay, so this is one fate of pyruvate where so this process is called fermentation. This is fermentation to alcohol and yeast. So this step is actually used in the ethanol production. So alcohol production for beverages or ethanol production in industries, we use yeast and yeast basically does this fermentation. So we grow the yeast under anaerobic condition without providing any oxygen, and under anaerobic condition it will convert the pyruvate to ethanol, and that's how ethanol is produced industrially. Okay, so this is one step. Second step is the one where pyruvate gets converted into lactate. So this actually happens in our body as well. So when you vigorously exercise, or when you run very fast, you feel a very strong pain in your muscle, right? That pain is actually due to the lactic acid formation or lactate formation, and that occurs under hypoxic or anaerobic conditions. So when we are running very fast, we tend to so our body cannot have enough amount of oxygen. Since we are running very fast, the oxygen supply is limited to our body, and our body then uh, is unable to follow this pathway where pyruvate is converted into acetyl coenzyme and further oxidation into CO2 and H2O. So this pathway cannot be followed, and that's when what our body does, our body starts converting the pyruvate into lactate. So that is also fermentation process, and in that process, lactate is formed, and that lactic acid actually causes the muscle pain that we feel while running very fast. Okay, so these are two fates of pyruvate under anaerobic conditions. That's clear to everyone? So whoever answered, all of you answered option B. So hopefully that's clear. Anybody has any doubt? Okay, great. Moving on to the next question then. What is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? What is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? So here the delta G value for the whole reaction that occurs starting from glucose and final product, that is CO2 and H2O. The delta G for that reaction is given as minus 686 kilocalorie per mole, and delta G of ATP is given as minus 7.3 kilocalories per mole. Okay. So you have to calculate the efficiency of ATP generation. So from one molecule of glucose, we are getting how many ATPs? 36 ATPs. Okay. And the delta G for one ATP molecule is given as minus 7.3 kilocalorie per mole. So how many kilocalories per mole of glucose will be there in total? So for 1 ATP, it is 7.3. So for 36 ATP, you can calculate that. And then you will get the delta G of ATP. And you have the total free energy value as 686. So you have to calculate the percentage of the free energy of ATP. Do the calculation. Try to do the calculation. So 1 molecule of glucose is giving 36 ATP. Okay. And for 1 ATP, it is taking 7.3 kilocalories per mole. The free energy is 7.3 kilocalories per mole. So for 36 ATP, it would be 36 into 7.3 kilocalories per mole. Okay. So that would be the value of free energy for ATP. Now, that what is the percentage of that if the total is if the total free energy is 686 kilocalorie per mole. So you have to find the percentage uh, of the free energy of ATP. So that will give you the efficiency of ATP generation. Anyone is able to solve this? So how do you find the percentage? Once you find the free energy of ATP, divided by the total and multiply it by 100. Okay, we have one answer in the chat box, 40. Okay, anyone else is able to solve it? You are closer to the correct answer. Okay, another answer is option B, that is 38. Okay, let's see the correct answer. Correct answer is 38% actually. So whoever got 40, you might have done some calculation mistake probably, or you have taken a decimal place wrong. So ATP generated from 1 mole of glucose would be equal to 36 into 7.3. That is 262.8 kilocalories per mole of glucose. Now, free energy of the complete glucose oxidation is given as 686 kilocalorie per mole. So, this is the complete reaction, complete oxidation of glucose. So, from glucose, 1 molecule of glucose, 6 carbon dioxide and 6 water molecules have formed. And the total free energy is 686 kilocalories per mole. Okay. So, now we have to just find the percentage. So, 262.8 divided by 686 into 100. That would give us the efficiency of ATP generation from 1 mole of glucose by respiration. And the answer is coming as 38% approximately. Is this clear? Anybody has any doubt in this? So you got 40 as the answer, I think. Where did you go wrong? I think you have done calculation mistake, or were you doing something completely different? Did you do the calculation like this? So whenever you are doing the calculations, be careful, because a small mistake can end up giving you the wrong answer. Yeah, just be careful from the next time, okay? Chalo, we'll move to the next question, question number 10. The electrons released by the citric acid cycle move down to the electron transport chain in which of the following manner? The electrons that are released by the citric acid cycle move down to the electron transport chain in which of the following manner? Option A is NADH, complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then H2O. Option B is complex 1, coenzyme Q, cytochrome C, complex 2, complex 3, complex 4, and then H2O. Option C is NADH, then complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then H2O. And option D is NADH, complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then H2O. Okay, C and D seems to be the same option. So answer between the three options. Tell me the series, how the electrons will be transported in the electron transport chain. What do you think would be the correct answer here? Anyone? Can anyone answer this question? Both A and C. Okay. Yeah, so that is the correct flow. So here you see uh, electron transport chain basically occurs. So you have glycolysis. From glycolysis, we were getting pyruvate, right? So those pyruvate molecules get converted into acetyl coenzyme A, and that acetyl coenzyme A enters the citric acid cycle. Okay. So from that citric acid cycle, NADH and FADH2 are generated, which are basically the electron which are carrying the electron. They will get converted into NAD plus and FAD plus respectively. So NADH here you can see gets converted into NAD plus and releases an electron. That electron, so from NADH, that electron flows to complex 1. So complex 1 is NADH dehydrogenase. It's an enzyme. It will carry that electron and it will transfer that electron to a coenzyme Q. Okay. Simultaneously, complex 2 will carry the electron from FADH2. FADH2 will get converted into FAD plus, and that electron will also be transferred to coenzyme Q. So now from NADH, the electron goes to complex 1 and then it moves on to coenzyme Q. From coenzyme Q, the electrons will move to complex 3. Okay. From complex 3, the electrons will move to cytochrome C, and cytochrome C will transfer the electrons to complex 4. From complex 4, these electrons will be accepted by the oxygen molecule. So I told you the terminal electron acceptor in case of aerobic respiration is oxygen. So these electrons will be accepted by oxygen molecules and the final product will be water. Okay, so oxygen will accept these electrons and there will be two protons. So the final product will be H2O. Okay. So electron flow is happening from NADH to complex 1 to coenzyme Q to complex 3 to cytochrome C to complex 4 and finally to H2O. Okay, this is clear. That's clear to everybody, right? And then after the flow of electrons, there's a so every time the electrons are transferred to one complex to another, some protons are pumped from the mitochondrial matrix to the intermembrane space, and that creates a proton gradient. So the concentration of proton becomes higher in this area than this area. You can see the protons are being pumped outside, and because of this proton gradient, the protons are then transferred to from the intermembrane space to the mitochondrial matrix by the fifth complex or the ATP synthase. So this blue uh, molecule that you see here that is embedded in the membrane, it is the ATP synthase, that is the main molecule, that is the main enzyme that converts or that synthesizes ATP, which is the most important molecule for the energy generation. So without ATP, none of the processes will be happening. So ATP formation is the most important step in all these metabolic processes. We are finally, so whatever we are eating, we are finally aiming to generate energy. Okay, so that's how we are going to live or we are going to survive. This is clear to everyone? Okay. Moving on to the next question. The conversion of ATP into ADP and PI, that is the inorganic phosphate, is an example of what type of reaction? The conversion of ATP into ADP and PI is an example of what type of reaction? Option A, oxidation reduction. Option B, amination. Option C, phosphorylation. Option D, hydrolysis. What type of reaction is happening during the conversion of ATP into ADP and PI? Oxidation reduction, amination, phosphorylation, or hydrolysis? ATP is getting converted into ADP and inorganic phosphate is released. So what type of reaction is happening here? Anyone? Okay, I have one answer, option C, phosphorylation. What about others? Okay, option D, that is hydrolysis. Okay, anyone else? Anybody else wants to answer? So here you see from ATP, ADP and inorganic phosphate is forming. That means phosphate is getting released from ATP molecule. So it cannot be phosphorylation. It is a dephosphorylation reaction. Uh, if the opposite was happening, you could have said that it is a phosphorylation reaction because the phosphate is attaching to the ADP molecule to form ATP. The correct answer is option D, hydrolysis. So the conversion of ATP to ADP is an example of hydrolysis reaction. This is how the ATP hydrolysis happens. So hydrolysis basically means the water molecule breaks down a bond and gets attached to that position. So you can see the lone pairs on the oxygen of water. They are attacking the phosphate here, and the phosphate, this bond is uh, breaking. And when this bond breaks, one hydrogen of the water molecule gets attached to this oxygen, and the remaining OH gets attached to the phosphate that is released. Okay, so this is the inorganic phosphate and this is the ADP. So from ATP, we are getting ADP and an inorganic phosphate. This is clear? Is this clear to everyone? Okay, let's move on to the next question. Question number 12. In eukaryotes, while generating ATP molecule from ADP, the ATP synthase translocates the protons from. So I showed you the electron transport chain, right? So I told you the protons are moved from one part to the another. So from where the protons are translocated from? Option A is cytoplasm to mitochondrial matrix. Option B is intermembrane space to the matrix of mitochondria. Option C is matrix to the intermembrane space of mitochondria. And option D is cytoplasm to the intermembrane space. So while generating ATP molecules from ADP, the ATP synthase translocates the protons from. What would be the correct answer? Write the answer in the chat box. What will be the correct answer? I've already shown you the diagram for electron transport chain. Anyone knows the answer? What would be the correct answer here? Okay. I have one answer, option B, intermembrane space to matrix of mitochondria. What about others? Anyone else? Okay, I have another answer, option D, cytoplasm to intermembrane space. Let's see the correct answer. The correct answer is option B, intermembrane space to the matrix of mitochondria. So this is the electron transport chain I showed you. So I told you that the protons are first pumped from the matrix of mitochondria to the intermembrane space by all these complex when the electrons are transported. Then finally, what happens? The concentration of protons in the intermembrane space is very high as compared to the mitochondrial matrix. This creates a proton gradient across the membrane, across the mitochondrial membrane. And because of this proton gradient, the ATP synthase now pumps all these protons in the intermembrane space to the mitochondrial matrix. And when it pumps a proton molecule that kind of drives the ATP synthase, that kind of drives the enzyme to form an ATP molecule from an ADP molecule. Okay. So protons drives the ATP synthase to function. Okay. So it's like a fuel to the enzyme. Okay. So if the enzyme is a car, protons are the fuel to that enzyme. Okay. So without the protons, the enzyme won't be functional. Without the proton pumping, the enzyme won't be able to perform its function. Enzyme won't be able to convert the ADP to ATP. Why does it need these protons? Why does it need a fuel to function? So the conversion of ADP into ATP, it's a energy consuming process. So the phosphate has to be attached to this ADP molecule. So the hydrolysis process of ATP is uh, energetically favorable. It can the H2O can easily attack the ATP, but the formation of ATP from ADP, this is a energy driven process, and it is an energy consuming process, and that energy is actually acquired by the pumping of the proton from the intermembrane space to the mitochondrial matrix. Is that clear to everyone? If you have any doubt in this, please ask. This is clear to everybody? If anyone has any doubt, please write it in the chat box. I'll try to answer it and clarify a doubt. Moving on to the next question, question number 13. Which of the following has the lowest E0 value? So it is the redox potential value, reduction potential. So which of the following has the lowest E0 values? Option A is ubiquinone, oxidized versus reduced. Option B is cytochrome C. Option C is NAD plus and NADH. And option D is oxygen by water, O2 by H2O. So ubiquinone, oxidized versus reduced, then cytochrome C, oxidized versus reduced, then NAD plus versus NADH, and option D is oxygen versus water molecule. Which of the following has lowest E0 values? Anyone knows the answer to this? Anybody? Okay, we have two answers, option D and option C. Let's see. Correct answer is option C, NAD plus and NADH. So this is the list of the E naught values of certain reactions, biological reactions that goes on inside our system. So you can see NAD plus and NADH, the redox potential is minus 0.3, whereas all the other reactions, that is ubiquinone, cytochrome C, and O2 by H2O, all have a positive value. So the lowest among all is NAD plus NADH. So that is the correct answer. That's clear to everyone. Moving to the next question. Which of the following statements is not correct about the electron transport chain? Which of the following statement is not correct? Okay, you have to identify the incorrect statement about the electron transport chain. So I've shown you the electron transport chain, so you should be able to answer this. Option A is coenzyme Q is also known as ubiquinone. Option B is complex 1 is also known as NADH dehydrogenase. Option C is cytochrome C accepts electrons from complex 2, and option D is coenzyme Q accepts electrons from complex 1 and 2. So which of the following is not a correct statement about electron transport chain? You all have seen the electron transport chain now, so everybody should be able to answer this question. Write the answers in the chat box. You have to identify the incorrect statement, okay? Keep that in mind and write the answer in the chat box. So we have three answers in the chat box, B, C, and A. So B is complex 1, also known as NADH dehydrogenase. C is cytochrome C accepts electron from complex 2. And A is coenzyme Q is known as ubiquinone. Okay. Anyone else wants to answer? The correct answer is option C. Cytochrome C accepts electrons from complex 2. That is the incorrect statement. So electron transport chain I have shown you. So this complex 1, I told you it is called NADH dehydrogenase. It basically reduces the NADH to NAD plus and takes the electrons. Complex 1 and 2 transfers the electrons to coenzyme Q. So that is the correct option. Option D is also correct. Coenzyme Q is known as ubiquinone. So that is also the correct answer. It is known as ubiquinone. Cytochrome C accepts electrons from complex 3. So that is the wrong answer, wrong statement here. Cytochrome C accepts electrons from complex 2. That is the option C. That is the incorrect statement because cytochrome C accepts the electrons from complex 2, as you can see here. Coenzyme Q is accepting the electrons from 1 and 2. Cytochrome C accepts the electrons from 3. Okay, and passes it on to complex 4. Complex 2 is also known as succinate dehydrogenase. Okay, like complex 1 is known as NADH dehydrogenase. Complex 2 is also an enzyme called succinate dehydrogenase. This is clear? Is this clear to everyone? If you have any doubt, please ask. Anybody has any doubt in this? Okay, if this is clear, I'll move on to the next question. Question number 15. The ATP synthase powers the generation of ATP molecule with the help of option A, ionic gradient generated by the movement of ions from the inner membrane to the matrix of mitochondria during electron transport chain. Option B, intercellular movement of phosphate ions during the electron transport. Option C, intracellular movement of ATP and ADP molecules. And option D, proton gradient generated by movement of protons between the matrix and the intermembrane space in the mitochondria during the electron transport. I've already discussed this, so everybody should be able to answer. ATP synthase powers the generation of ATP molecules with the help of. How does the ATP synthase gets the power? How does the ATP synthase powers the generation of ATP molecules? Is it ionic gradient generated by the movement of ions? Is it intercellular movement of phosphate ions? Is it intracellular movement of ADP and ATP? Or is it proton gradient generated by the movement of protons? What is the correct answer? This I have discussed. I have told you all of you the correct answer in the previous question already. So everybody should be able to answer. I want answers from everyone. Write the answers in the chat box. What is the correct answer here? Come on. This should not take up much time. Come on, write your answers in the chat box. What would be how is ATP synthase generating the ATP molecules? Nobody wants to answer. What is the thing that was moving across the membrane because of which ATP was getting synthesized? I have one answer in the chat box. Out of 20 people, only 2 answers are there in the chat box. I've shown you this question. I've told you the answer to this question already in the previous question, previous explanation. Option D is the correct answer. Good to all of you who did write the answer. So I already have explained the protons are moved from the matrix to the intermembrane space that creates a proton gradient. The gradient means the concentration of protons uh, is not equal. It is higher in one side of the membrane and it is lower at the other side of the membrane. So it is kind of uh, creating a gradient of the concentration from higher to lower, and thus it is moved from the higher concentration to the lower concentration area, so that protons are transported from the intermembrane space to the mitochondrial matrix by the ATP synthase. It pumps the protons and uses that as the power to generate ATP molecules from the ADP molecules. I hope that's clear. People who are not writing the answers, is it clear to you? I'm assuming it's clear to you. That's why you are not asking any questions. If you have doubts, if you are not able to understand this, please ask. This is a doubt clearing session. I am here to clear your doubt. Please ask your doubt, otherwise I won't be able to clarify it. I'm assuming it's clear to everyone since no one is asking anything. Moving on to the next question. Which of the following is a terminal electron acceptor for aerobic reactions? Which of the following is a terminal electron acceptor for the aerobic reactions? I have told you this also in the previous question. So electrons are transported in the electron transport chain and finally accepted by one molecule. What is that molecule? Option A is Fe3, option B is O2, option C is sulfate, SO42 minus, and option D is nitrate, NO3 minus. Which molecule acts as a terminal electron acceptor in case of aerobic reactions? Write your answers in the chat box. What is the terminal electron acceptor for an aerobic reaction? The answer is there in the question itself. Read the question carefully, you will be able to answer this. Everybody write the answer in the chat box. Okay, I have two answers, option C and option B. So in the question itself, you can see it is saying aerobic reactions. That means in the presence of oxygen. So the terminal electron acceptor in such cases is oxygen. Okay. So option B is the correct answer. In case of aerobic reaction, oxygen is the terminal electron acceptor. All the options, other options are terminal electron acceptors in case of anaerobic reaction where oxygen is not present. In that case, all these other options act as the terminal electron acceptor. So you can see here, so if it is an aerobic respiration, the terminal electron acceptor is oxygen. And phosphorylation for ATP generation is basically substrate level phosphorylation and oxidative phosphorylation. So SLP stands for substrate level phosphorylation and this stands for oxidative phosphorylation. And total 38 molecules of ATP is generated. So by oxidative phosphorylation, 36 molecules are generated and substrate level phosphorylation occurs in glycolysis. So I told you in the 10 steps of glycolysis, we were getting 2 molecules of ATP, right? So those steps where the ATP was formed, ATP was generated in the glycolysis pathway, those processes were called substrate level phosphorylation because the ATP is generated from a substrate, okay? And the other molecules of ATP that are generated by the ATP synthase by by that proton pump, that is the oxidative phosphorylation pathway. Uh, because in that case, the ATP is directly formed from ADP. The phosphate is attached to the ADP by the proton pump powering the ATP synthase. So that is a oxidative phosphorylation where the phosphorylation is happening because of the oxidative pathway, because of the oxidation that is happening when the oxygen is accepting the electrons from the electron transport chain, because of which proton gradient is formed and that proton gradient drives the ATP synthase. But in glycolysis, the two molecules of ATP that are forming, that is called substrate level phosphorylation because directly from a substrate that phosphate is transferred to the ADP molecule. Okay, that is substrate level phosphorylation. So in aerobic respiration, oxygen is the terminal electron acceptor. In anaerobic respiration, iron, manganese, sulfate, nitrate, all these can be the terminal electron acceptor depending on the organism and type of respiration. And again, ATP generation can be both substrate level and oxidative phosphorylation. Just in case of oxidative phosphorylation, it would be uh, sulfate or nitrate that will accept the terminal electron instead of oxygen. Okay. And the number of ATP can be anywhere between 2 to 38 depending on the organism. And then there's third pathway called fermentation pathway, which I have discussed already, where the which is also an anaerobic anaerobic process, but in that case, the terminal electron acceptors are generally organic compounds. So you remember the fermentation that was occurring in yeast that was converting the pyruvate into ethanol and carbon dioxide. So that those are the fermentation processes, and there the phosphorylation for ATP generation is only substrate level because only the glycolysis part is generating the ATP and only two molecules of ATP are generated. So for fermentation is not a process where a lot of energy is produced, since ATP is the key energy energy generating molecule. So more the ATP, more energy will be there in an organism. Okay, so aerobic respiration clearly generates more number of ATPs than any of the other processes. This is clear to everybody? Anyone has any doubt in this, please ask. If you have any doubt, please write it in the chat box. I'll move on to the next question. Question number 17. Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzymes? Oxidation of N-octane to acetyl coenzyme E. Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzymes? Option A, phosphorylase. Option B, monooxygenase. Option C, lipase. Option D, catalase. Anybody knows the answer to this? Write it in the chat box. Oxidation of N-octane to acetyl coenzyme A is catalyzed by which of the following enzymes? Phosphorylase, monooxygenase, lipase, and catalase. Write your answers in the chat box. Anybody? Okay, the correct answer to this is monooxygenase, option B. So this is how the pathway goes, and this is basically a process uh, that leads to beta oxidation. And beta oxidation is a process to catalyze the fatty acids. So these organic, so this is alkane, right? So it's a 8-carbon long chain. Uh, so this octane is catalyzed, it is converted into an alcohol, octanol. Then it is converted into aldehyde, octanol, and then finally to carboxylic acid, that is octanoic acid, and that carboxylic acid then is converted into acetyl coenzyme A by beta oxidation. So the first step, octane to octanol, it is catalyzed by monooxygenase. So the first oxidation step, that is converting the alkane into alcohol, it is catalyzed by monooxygenase. Is this clear? If you have any doubt, please ask. Okay. So the next question is, the steps of anaerobic degradation of alkene follows which of the following steps? Option A, alkene to aldehyde to acetate. Option B, alkene to alcohol to acetic acid. Option C, alkene to alcohol to aldehyde to carboxylic acid. And option D, alkene to aldehyde to alcohol to carbon dioxide. Just showed you the pathway. So tell me what would be the correct answer. The degradation of alkenes, how will it go? Alkene to aldehyde to acetate, or alkene to alcohol to acetic acid, or alkene to alcohol to aldehyde to carboxylic acid, or alkene to aldehyde to alcohol to carbon dioxide. Just showed you the beta oxidation pathway, right? So how is it going? Write your answers in the chat box. Option C, alkene to alcohol to aldehyde to carboxylic acid. Okay, so two people are answering option C. That is the correct answer. So it is the alkenes are going to alcohol first, and then it is getting converted into aldehyde and then carboxylic acid. So that is how and from carboxylic acid beta oxidation pathway can go on, and acetyl coenzyme A will be formed. Okay, so that is the correct answer. Moving on to the next question. Synthetic organic chemicals found in the soil that do not occur naturally are called option A, antibiotic, option B, xenobiotics, option C, chemobiotic, and option D, all of the above. We have already discussed this, so this should be easy to answer. Synthetic organic chemicals which are found in soil that do not occur naturally. Compounds that are synthesized, that are not occurring naturally, they are called option A, antibiotic, option B, xenobiotic, option C, chemobiotic, or option D, all of the above. Write your answers in the chat box. We have discussed this in the start of the session. The first 2 or 3 questions were related to these compounds. So what is the correct answer? Okay, I have one answer in the chat box, option B, that is xenobiotic. That is the correct answer. So I've already discussed xenobiotics are the synthetic organic compounds that are not naturally available. Okay. Next question, match the following enzymes with its substrate. So you can see there are 4 enzymes of the uh, glycolysis pathway: hexokinase, pyruvate kinase, enolase, and aldolase. And there are 4 substrates given. First is fructose 1,6 biphosphate. Second is 2-phosphoglycerate. Third is phosphoenol pyruvate. And fourth is glucose. So match the enzymes with its substrate. So remember the glycolysis pathway. Come on, write the answers in the chat box. What would be the substrate for hexokinase? What would be the substrate for pyruvate kinase? Then enolase and aldolase. Okay, I have one answer as option A. Okay. What about others? Anyone else wants to answer? Anyone else knows the answer? Or anyone else is able to solve this question? So I'll start with the first enzyme, that is hexokinase. That is the first enzyme in the glycolysis pathway also. So that is your hint. Now think about the answer. Match the enzymes with its substrate. Hexokinase is the first enzyme in the glycolysis pathway. So what would be the substrate for hexokinase? What is the first molecule from which glycolysis pathway starts? What would be the substrate of hexokinase? Just think of that. Anybody? Can anybody tell me what would be the substrate for hexokinase? Hexokinase is the first enzyme in the glycolysis pathway. So glycolysis pathway starts with which compound, which molecule, which substrate? Come on, write the answer in the chat box. I told you glycolysis starts with, it starts with glucose, right? So the substrate for hexokinase would be glucose, right? So if the substrate for hexokinase is glucose, that means 1, 4. We have two options here, C and D. Then second enzyme here is pyruvate kinase. So pyruvate kinase, the substrate for pyruvate kinase is phosphoenol pyruvate. So basically from phosphoenol pyruvate, phosphate is transferred to the ADP molecule by the enzyme pyruvate kinase, and from phosphoenol pyruvate we get pyruvate. This is the last step of glycolysis. So 2, 3, and then enolase, substrate for enolase is 2-phosphoglycerate. From 2-phosphoglycerate we get phosphoenol pyruvate, and then last is aldolase. The substrate for aldolase is 1-fructose 1,6 biphosphate, which gives us two products, glyceraldehyde biphosphate and dihydroxyacetone phosphate. So option D is the correct answer here. So the pathway starts from glucose. From glucose, it goes to glucose 6-phosphate by the enzyme called hexokinase. Hexokinase or glucokinase. So hexokinase can actually convert other hexose sugars also. So that's why the name is generalized to hexokinase. So uh, glucose is the substrate for hexokinase. Then from glucose 6-phosphate, it is converted to fructose 6-phosphate by a glucose 6-phosphate isomerase. So that for that enzyme, glucose 6-phosphate is the substrate. Then phosphofructokinase. For phosphofructokinase, fructose 6-phosphate is the substrate and fructose 1,6 biphosphate is the product. Then aldolase uses fructose 1,6 biphosphate as the substrate and converts into two products, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then these get converted into 1,3 biphosphoglycerate by glyceraldehyde phosphate dehydrogenase. Then phosphoglycerate kinase uses 1,3-biphosphoglycerate as the substrate to form 3-phosphoglycerate. Then mutase converts 3-phosphoglycerate into 2-phosphoglycerate. Enolase uses 2-phosphoglycerate as the substrate to convert into phosphoenol pyruvate. And then phosphoenol pyruvate is the substrate for pyruvate kinase to form the product pyruvate, final product of glycolysis. Okay. This is clear to everybody. Moving on to the last question of this session. Question number 21. How many reaction that occurs in TCA cycle transfers electrons from one substrate to an electron accepting enzyme? How many reactions that occurs in the TCA cycle transfers electrons from the substrate to the electron accepting enzyme? So this is TCA cycle is the citric acid cycle. So I told you after glycolysis, we have citric acid cycle where the pyruvate gets converted into acetyl coenzyme, and that acetyl coenzyme enters the citric acid cycle, and it generates basically NADH and FADH2, that are the electron accepting enzymes. These are the compounds that will then go on to the electron transport chain and transfer the electrons. Okay. So how basically you have to tell how many steps are there in the TCA cycle where NADH or FADH2 is generated. I have one answer in the chat box. 4. That is the correct answer. So there are 4 steps in the citric acid cycle. So here you can see acetyl coenzyme A is the entry point, is the starting point of the citric acid cycle. It enters the cycle, gets converted into citrate, aconitate, and then isocitrate, alpha ketoglutarate. So when the conversion of isocitrate to alpha ketoglutarate happens, and then alpha ketoglutarate to succinyl coenzyme happens, 2 molecules of NADH are released, and then 1 molecule of FADH2 is released from succinate when it is converted to fumarate by succinate dehydrogenase. The last molecule of NADH is released from malate dehydrogenase, where malate is converted into oxaloacetate. So 3 NADH and 1 FADH2 are released. So total 4 molecules are released, and these NADH and FADH2 then will go to the electron transport chain. So basically in our body, we are whatever food we are eating, finally it will be broken down into glucose. That glucose will enter the glycolysis pathway. In the glycolysis pathway, glucose will get converted into pyruvate. Pyruvate or pyruvic acid will get converted into acetyl coenzyme A. That acetyl coenzyme A will enter the citric acid cycle, where it will generate NADH and FADH2, and the cycle will keep on going, and it will keep on producing more NADH and FADH2. These NADH and FADH2 will then enter the electron transport chain that I've explained earlier, where they will transfer the electron to all the complexes 1, 2, 3, and 4. While that electron transport occurs, a proton gradient is created across the mitochondrial matrix. Because of that proton gradient, the ATP synthase can now pump the protons, use that as the power, use that as the fuel to generate ATP, and that's how we are getting the energy from the food we are eating. That's clear to everybody? So this brings us to the end of the session. So if you have any doubt, please ask. I hope that's clear to everyone how all these pathways are interlinked to each other. So from glycolysis, we are moving to the citric acid cycle, and from citric acid cycle, we are moving on to the electron transport chain, and final ATP generation is happening. So you have seen the oxidative uh, aerobic respiration reaction where the glucose was getting converted into carbon dioxide and water. So this is how the process is going on. Glucose will get converted into pyruvic acid. From pyruvic acid, you will have carbon dioxide and water generated from the citric acid cycle and the electron transport chain. Okay, if everything is clear, we can end this session now. Thank you everyone for joining. I'll end the session now. Thank you.